Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Methods and Best Practices for the Development of Single Cells into Colonies. My name is Georgina Wynne-Hughes and I'll be moderating today's presentation. I'm delighted to be joined by our guest speakers, Dr. Jessica Hartman, Senior Director of Product Applications at Cell Microsystems, and Dr. Lindsey Kuehm, Field Applications Scientist at Cell Microsystems. In this webinar, we will be covering a series of case studies highlighting how scientists struggling with generating clones were able to overcome common bottlenecks in their research. Labor burden, poor cell viability, low efficiency, and lack of clonality are just some of the hurdles these investigators faced in developing the cell lines that are necessary for downstream discovery. Following the presentation, we will have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these at any time to the right of your screen. Without further delay, I would like to hand over to our speakers, and I would like to thank them again for presenting for us today.
Hello, my name is Jessica Hartman, and I'm the Senior Director of Product Applications. And I'd like to thank you for taking the time to join us today to hear about methods and best practices for the development of single cells into colonies and the challenges and solutions we're going to present to you today. So from this webinar, we hope you're going to take away some key learning objectives. Firstly, we want you to understand the different methodologies that are available to you for developing single cells into colonies, how to increase the efficiency of your single cell workflows, how to get more colonies from these workflows, but most importantly, how to get high viability cells that are exactly the phenotype that you need to advance your research. So if you're joining us today, I'm sure you're familiar with this process of monoclonal cell line development, but it really is the cornerstone and foundation of a vast array of research and development efforts, including cell and gene therapy, recombinant protein production, personalized medicine, drug discovery and toxicology, and generalized research and development for discovery. The entire workflow starts typically from this first step here, where you have a heterogeneous bulk population of cells that you've transfected with some kind of construct of interest, whether it's a knockout or a knock-in. And then you have to select for cells that stably express your gene of interest using a drug selection marker or a fluorochrome or a phenotypic expression marker. And then you have to screen and select single cell clones from that bulk population in order to make your monoclonal population. If you're lucky enough to get some single cell clones out of this workflow, what follows is extensive rounds of cell characterization, clonal expansion, and evaluation. And typically this process can take from weeks to months if it's not particularly efficient. But the most important step in this entire workflow is this step right here, where you have to screen and select your single cell clones. And this is really where all of your troubles begin if these workflows are a challenge for you. So we're going to launch our first poll question here. So please answer the questions and tell us what you are currently using for cell line development. So when you're in the cell culture lab, you spend all of your time, energy, and effort making sure that your bulk cell population is happy and healthy in its flask-like environment, whether it's in a tea flask or a plate. Cells are happiest when they are grown together in bulk culture. But in order to perform this complicated monoclonal cell line development workflow, the first step is to take those happy polyclonal cells, trypsinize them or dissociate them into a tube and turn them into a single cell suspension. And this is really where a lot of the issues with single cell cloning begin. There are many technologies that are available for single cell cloning that help you turn this population of cells into single cells in a well. They can range from the simple, such as limiting dilution, to the complex, such as high speed cell sorters shown on the right. There's a trade-off in these methodologies from labor to cost. It's very labor intensive to do manual limiting dilution, but it's not particularly expensive from a reagent or equipment standpoint, whereas these high-end sorters can cost you a lot of money, but they significantly reduce the labor involved. Interestingly, the current industry standard for these workflows has really focused mostly on isolating single cells with fluidic handling, whether that's droplet dispensing in low pressure sorters like you see in the middle, or high speed sorting with these high end sorters like you see on the right. Everything is focused on turning a single cell in suspension into a single cell in a well. But what's critical about this is that these methods ultimately remove the cells from all of the advantages of the flask-like culture environment, namely shared media, the benefits of cell-cell communication, and the paracrine signaling that you get in that particular environment. Unfortunately, there are well-known reductions in viabilities associated with this method. No matter how gentle you are, the cells ultimately end up in a well by themselves, and that limits their ability to propagate and form a happy, healthy colony. And unfortunately, there continues to be limited development of these non-fluidic sorting technologies, as the gold standard tends to be putting a single cell in a well in order to ensure monoclonality. If you're lucky enough to have one of these technologies that's really high efficiency and you're very lucky, you might end up with something that looks like this, where you see a single cell in each well of your 96 well or 384 well plate. But to do that, you had to use multiple pieces of equipment, and it's very laborious and manual manipulation. Most likely what you're going to see when you look into your plate after single cell dispensing is something that looks more like this, where most of your wells are empty because the cells have either not deposited or not survived, or you're going to see a few sparse single cell wells and perhaps some wells that have more than one cell in them. And unfortunately, you're mostly looking at stressed, non-viable cells that are going to struggle to provide you a happy, healthy colony. In addition, this is an extremely inefficient process, particularly for finding rare edits or rare variants in your population, largely because your starting population is so low. If you're only starting from a handful of wells that have a single cell in the well, the odds that you find that needle in a haystack go incrementally down. And as you can see where that little red X is, the other main issue that plagues monoclonal celling development is indeed that lack of clonality. It's incredibly difficult with these methodologies to ensure that you actually deposited a single cell, much less that that single cell formed a clone. And there's a lot of back-end analysis that's required in order to confirm that you started from a single cell, which is absolutely required for most of these workflows. We're now going to launch poll question number 2. So please answer and tell us what main issues face you in your single cell cloning workflows. So as I've already told you, the benefits of cell culture are really hinging on the fact that the cells are growing in a flask where they're able to communicate with each other and share paracrine signaling. But this does not allow for single cell spatial segregation that is required in order to do monoclonal celling development. So an ideal solution to solve these pain points in celling development would be a tissue culture consumable that combines the ability of the cells to communicate with flask-like culture, but also maintaining single cell spatial segregation. So today we're going to talk to you about the CellRaft array, which you can see on the left-hand side here. It's a specialized tissue culture consumable that allows you to seed your cells into that intercenter reservoir as you see here. It comes in multiple formats. It's very easy to use, just like any other tissue culture dish. And importantly here, all of the cells that you're seeding in that reservoir share a contiguous media, as you can see in the cartoon here. But uniquely, within the CellRaft array is fabricated thousands to tens of thousands of elastomeric microwells. This provides spatial segregation for the cells that you seed, such that every cell that goes into the array itself settles by gravity into an individual microwell, allowing for shared media, but also spatial segregation. Within each microwell is fabricated a polystyrene cell raft, where the cells can attach to that polystyrene growth surface, just like they would any other tissue culture dish, and grow normally on the surface of that raft. So what you can see in the cartoon in this cross section here is that when you seed your cells of interest in the CellRaft array, they settle into the elastomeric microwell onto that magnetic polystyrene cell raft, where they can attach and grow as adherent or suspension cells. And importantly, all of those microwells are sharing a contiguous media volume, so we maintain that shared media while also allowing for single cell spatial segregation and clonal formation. So I'm going to turn the webinar over to my colleague, Lindsey, who's going to tell you about some case studies that we've been working on that demonstrate how these issues can be overcome in your single cell workflows.
Hello, everyone. My name is Lindsey Kuehm, and I am a field application scientist. I'm going to introduce you to a few case studies that I worked on personally in-house with outside researchers and how we help them overcome the single-cell workflow bottlenecks. These cases include an adherent cell line that did not survive being sorted, a pharma company looking for a more efficient way to track clonality, and overcoming the struggles of iPSC cloning. As I'm sure you know, Many edited and even parental lines do not enjoy being sorted or isolated alone in a well. As Jessica mentioned, traditional methods lead to low cell viability and make it difficult to derive healthy clones that meet the researchers' exact criteria. We had a researcher working with a fast-growing murine intermedullary collecting duct cell line that had these struggles. They had a large, somewhat toxic piggyback transposed gene and attempted for a year to get a healthy gene-positive monoclonal population. For them, traditional sorting and limiting dilution methods led to low viability, poor colony formation, and then they had no monoclonal gene-positive populations after these methods. This researcher clearly needed a more adherent, cell-friendly, and efficient way to get the clone they desired because the process they were trying was too time-consuming and lacked project progression. To streamline their workflow, we were able to condense the entire workflow into one consumable, the CellRaft array. We seeded their cells onto the array, let them settle and adhere for 24 hours, and then induced gene expression with doxycycline. 24 hours after gene induction, we stained for gene expression directly on the array and used our system to visualize gene-positive monoclonal populations. After that, we were able to identify hundreds of potential targets and isolated 124 clones, 105 of which showed outgrowth after isolation. Here you can see the progression of a single cell into a gene-positive clone on the array and the clonal outgrowth after isolation. So on day 0, when we seeded the cells, we allowed them to settle, and then we took that first scan to capture the wells that had a single cell. On day 1 after seeding, we imaged the cells again on array, identified dividing cells, and then were able to induce gene expression directly on the array. On day 2 after seeding, we stained for the gene of interest directly on the array, and then imaged the cells for colony formation and the positive gene expression. Then after identifying the monoclonal gene-positive colonies, we isolated 124 populations and monitored them for outgrowth. We were then able to send 10 clones to the researcher so they could confirm the clonality and the presence of the gene of interest. They performed a Western blot as well as IHC staining to confirm expression of the gene of interest. All 10 clones were positive for the gene that they screened on the CellRaft array, and shockingly to the investigator, one of the 10 clones happened to be double positive for a second gene that had been transfected that was not fluorescently labeled and not screenable. There are 2 key takeaways from this case study about single-cell cloning. First, simply in eliminating fluidic manipulators, the single-cell isolation, we were able to overcome a year-long investigator's nightmares with cell line development in a week. Second, by dramatically increasing the number of available monoclonal colonies, the chance of identifying a single or double positive clone was significantly higher, enabling the researcher to find his elusive clone and move his research forward. In the case of single-cell workflows, efficiency and clonal assurance are key. In this case study, the cell line engineering core at a pharma company was tasked with generating hundreds of monoclonal cell lines from both adherent and suspension cells for internal users. Despite having access to several high-end flow sorters, a bottleneck for them was cell survival post-sorting and the inability to confirm clonality post-single cell sorting. Their workflow is depicted here and typically took 12 to 18 weeks to identify a suitable clone using flow sorting. To improve their success rates and speed the time from project initiation to clonal development, they compared several methods of bulk sorting and/or resorting cells after an initial bulk sort. They compared these instruments for ease of use, efficiency, track and traceability, and also cell viability. As you can see here on the CellRaft array, the researchers were able to easily track and trace from a single suspension cell into a monoclonal population, and then isolate colonies of interest for clonal expansion. They found that they could use the CellRaft AIR system even without performing an initial bulk sort. The key takeaways from this case study are that the lack of clonality, poor cell survival, and inefficient workflows continue to be bottlenecks for high throughput research and discovery. By utilizing the CellRaft array, the cell line development time decreases from months to weeks. No expensive secondary confirmation of clonality is necessary, and the ease of use is dramatically improved compared to traditional bulk sorting methods. As some of you already know, a good, healthy iPSC is very difficult to find. Traditional iPSC workflows are plagued by challenges that limit their utility in research and development efforts. These challenges include an inefficient process, poor viability of iPSCs, lack of trace and trackability of clonality of these iPSCs, having to use very expensive media and additives, loss of pluripotency during expansion, and also there's only manual isolation methods for these iPSCs. The flash-like conditions of the CellRaft array and the automated imaging and isolation using the CellRaft AIR system allows for track and traceability from a single cell into a clone, the ability to screen more cells, so 500 times more cells per consumable. It's compatible with any iPSC coding, and you're going to use 1,000 times less cell coding. So you can see here on the right where we used 4 different iPSC coatings and 4 different cell lines, and you can see the healthy, happy growth of these cell lines from a single cell into a colony. It is also compatible with any iPSC media, and here you also use 2,000 times less media per cell screened. Often while in culture, iPSCs will begin to differentiate and they are no longer viable for the downstream application. Pluripotency is maintained on the CellRaft array during culture and after isolation, and is easily confirmed by staining for pluripotency markers such as TRA160 on and off the array. So your iPSCs stay iPSCs and you can track a monoclonal population of pluripotent cells. We directly compared colony formation of iPSCs using limiting dilution with the CellRaft AIR system. As you can see here, the CellRaft AIR system allows for the easy identification of a single cell clone, and the flash-like conditions allow for healthy growth of viable colonies. Using 2 different iPSC coatings, we showed that in one consumable, we were able to obtain 213 clones on iMatrix, and then also 262 clones using the HESC Matrigel. These are the total clones in one consumable. And using one consumable for limiting dilution, which is a 96 well plate, we were only able to get 10 total clones. When we looked at the percentage of outgrowth on these 2 matrices from a 96 well plate that we isolated from the CellRaft array, we had 82% and 73% of our colonies grow off of the raft and form healthy clones. While during limiting dilution in 196 well plate, we only had 10% of our clones that formed viable colonies. There are a myriad of iPSC workflows that can be performed directly on the array itself, allowing the researcher to use just one consumable, such as reprogramming fibroblasts into iPSCs and forming monoclonal populations. You can simply expand your iPSC cell line. You can differentiate iPSCs into organoids directly on the array, and you can also characterize your iPSCs using immunofluorescence. But today I'm going to focus on differentiation of iPSCs into organoids directly on this CellRaft array. So here we have human iPSCs that have been differentiated into cerebral organoids that continue to grow and form neural networks after isolation. So here you can see the single cell on day 1, the single iPSC. We induced embroid body formation, induced neuronal induction, and then expanded these organoids on the array for 10 days. After they matured, we isolated them and let them continue to grow. And as you can see, by day 37, those neuronal networks are starting to form after isolation. So with the CellRaft AIR system, a single iPSC can be easily identified and monitored for clonality. The culture conditions improved clonal iPSC generation by 25 times compared to limiting dilution. Pluripotency can directly be confirmed prior to clonal expansion. There's a significant reduction in time, consumables, materials, and also labor required for iPSC clonal development, and it is compatible with 2D and 3D applications. So using our technology, we are able to significantly reduce the consumable and reagent costs associated with these workflows, improve cell survival and maintain pluripotency, and automate a process that is typically completely manual and requires an expert scientist to execute. In addition, the favorable growth conditions allow us to perform 2D and 3D works in a single consumable. And now we will pause for poll question number 3. Please feel free to answer.
Thank you, Lindsey, for that really interesting discussion of how you were able to help investigators advance their research by using the CellRaft array. So now that Lindsey has introduced the technology to you and shown you how improving culture conditions for cells in the CellRaft array can improve and speed the time to cell line development, I'm sure you're asking yourself, how does this technology actually work and how can I use it in my lab? So our solution comprises 3 key components of the CellRaft technology. First is the CellRaft array, which you've already heard some about earlier in the webinar. That is our custom tissue culture consumable, which maintains flask-like culture conditions while allowing for single cell separation, and then ultimately culturing your cells on that small polystyrene CellRaft you see on the inset. But the CellRaft array is only as powerful as the technology that supports it. And that is the CellRaft AIR system, which is our instrumentation that allows for automated scanning of the CellRaft array, as well as acquisition of those CellRafts that you are interested in. In addition, nobody wants to search through thousands of rafts looking for your clone of interest, and that's where CellRaft cytometry comes in. It's our proprietary software that allows for identification of single cells, and then you can monitor those single cells into a clone of your desired choosing. And then importantly, it's not just the ability to identify and find these cells, we also have the ability to use the AIR system to automatically retrieve high viability cells, colonies, or organoids that can then be propagated for your workflows downstream in a 96-well plate. So to give you a bit more information about each of these key pieces of the workflow, I'd like to tell you a little bit more about the CellRaft array itself. As I mentioned previously, it's a novel proprietary culture dish that contains thousands of microscale cell culture growth surfaces called CellRafts. Each CellRaft is housed within an individual microwell, which allows for gravity-based separation of your bulk population into single cells by a Poisson-like statistical distribution. The CellRafts themselves are optically transparent, allowing for high-resolution imaging of the cells on the rafts, and those rafts are releasable from the array. But most importantly, as we've talked about, that flask-like culture environment with the shared media is really the key to the whole workflow because it allows for improved viability and proliferation of the cells that you're screening. Once you have seeded your cells onto the CellRaft array, you can image that array itself on our instrument, the CellRaft AIR, shown here on the left. The instrument is fully automated and allows you to image in bright field and 3-color fluorescence with blue, green, and red fluorescence. We can identify rafts of interest with a 10x magnification, and those images allow us to perform in-depth software analysis in order to identify the clones of interest for your downstream workflows. As you can see in the inset here, the CellRaft array sits on the left-hand side of the deck, and your collection plate sits on the right-hand side of the deck. But importantly, the software really is the key to enabling these high efficiency workflows to identify the cells or colonies that are of interest to you. As you can see here in this cartoon on the left, your population of interest is very likely to be heterogeneous. You might have cells that are single positive, double positive, or not positive at all for your gene of interest. And you don't want to go looking for those rare double positive cells in your CellRaft array. So how do we find them using our software? With one mode, you can do CellRaft review, which allows you to interact with every single field of view in the CellRaft array and look at every single CellRaft over time. This is very manual, and it is a great option if you want to look at all of your cells. But if you, like most researchers, have deadlines to meet and experiments to perform, CellRaft cytometry is going to be your best friend. It allows you to look at every scan that you've performed on the array, to identify specific user-defined parameters to identify the cells that you want based off of size, shape, morphology, or fluorescence, and then define those populations for identification of cell rafts containing cells of interest. Once you've narrowed that population down, you can use CellRaft Organizer, which allows you to filter these populations based on any other criteria that you might like. To show you what that software looks like, here's a screenshot of the GUI from inside of the software. It uses advanced bright field analysis to identify cells of interest, meaning you don't have to have a fluorescent label to identify your cells. It allows you to define unique subpopulations shown here on the left that are user-defined and specifically tailored to your cell or application of interest. You can create subsets of these particular features to identify features over time that are important to you. So for example, in the example that I'm showing here, we can look at a variety of parameters and identify the intersect of them in order to create our population of interest. What I'm showing here is that in panel A, we are looking at single cells that are round at time point 1, cells that have doubled on the CellRaft at time point 2, and cells that have formed a colony at time point 3. And what we're interested in is the intersect of those three populations, namely for a single cell workflow, a CellRaft that had a single cell at time point 1 that divided and formed a clone at time point 3. Using CellRaft cytometry, we can easily identify these monoclonal derived populations, and then we can map them for isolation using the CellRaft AIR. So what does mapping them for isolation actually mean? Well, the last key feature of the technology is that we aren't just culturing the cells with this spatial segregation. We're also able to retrieve them, attach to the cell raft, and deposit them for isolation and expansion into a 96-well plate for growth. The AIR system allows you to map your rafts that contain colonies of interest to the plate. And then the CellRaft AIR system will perform automated CellRaft release and transfer to retrieve those colonies that you have specifically selected. So how does this work in the AIR system? The CellRaft array sits on the deck of the instrument, positioned above a release needle on the 10x objective. And then a collection wand that is magnetic dips into the fluid of the array to retrieve that released polystyrene cell raft. As I mentioned, the rafts are indeed releasable and retrievable. Your cells remain on the raft during the entire process of isolation. There's no need to trypsinize them or remove cells, and they can then be picked up with that magnetic wand because the cell rafts themselves have been doped with magnetic nanoparticles. To show you what this looks like in motion, on the actual instrument, as you can see here in this cartoon, once you've identified a cell raft containing a cell of interest, the release needle will actuate through that elastomeric microwell. It will pop out the cell raft. The wand will dip into the media, pick up the raft with your cells still attached, and gently deposit that raft containing your clone into media in your 96-well plate. At this stage, the cells can passively grow off of the cell raft to form all of those happy, viable clones that Lindsey showed you previously in the case studies that we demonstrated. Importantly, we've been able to demonstrate broad cell line compatibility with our technology across a wide variety of cell types. In-house, we have currently characterized over 80 different cell lines on the array with equal success. This includes primary cells such as iPSCs, embryonic stem cells, organoids. It includes your typical adherent cells such as 293Ts and CHOs, as well as other adherent cells like some cancer cell lines. And we can also do this with suspension cells as demonstrated in the pharma workflow, including cells such as Rajis and Jurkats. Importantly, the flexibility of the cell line AIR system allows you to utilize the technology for a wide variety of applications. While we've focused on cell line development today, it's been leveraged for genomics, for CRISPR gene editing, for stem cell expansion, for 3D growth, as well as other on-array applications such as cell-based assays. So to summarize, we would like to help you enhance your cell line development using the CellRaft AIR system. And hopefully today we have convinced you of the advantages of maintaining your healthy cells in contiguous media to improve single cell clone formation. We've shown you how we can automatically screen thousands of single cells on a single consumable to recover hundreds to thousands of clones. We're able to track single cell growth into a desired clone or organoid, and then using the AIR system, we can easily isolate single cell clones or organoids for downstream workflows. And ultimately, this allows you to simplify your workflows and have confidence in your downstream applications that the cell lines you've actually developed are fit for purpose and are ready to go in your downstream research. We'll pause here for a question, and then with that, Lindsey and I would like to conclude and thank you for your attention and your time, and we are happy to take any questions that you might have about the AIR system or the case studies presented here.
Thank you for that very informative presentation, Jessica and Lindsey. It's now time for our question and answer session. So just a reminder, if you do have any further questions, you can still send those in now. So you can submit these by typing into the questions box just to the right of your screen. So the first question we have is, how many clones can I expect to get with the CellRaft instrument?
Thanks, Georgina. Typically, with your most average cell line development workflows using some of your workhorse cell lines, you can expect hundreds to thousands of clones, even with rare or difficult to use cell types. In general, I would say the average that most of our users isolate off of the AIR system is around 96 to 120 or so. Most folks find that if they isolate a single 96-well plate, that because of the efficiency of outgrowth of our clones, your barrier to success is much, much lower. So of the 96 clones that you pick off of the AIR system, we fully expect greater than 80% of them to be viable. So most of our users report that they like to isolate between 1 and 2 96-well plates per experiment, but you could, of course, isolate many, many more if that was necessary for your workflow.
Okay, thank you. And second question we've got here is, I work with a variety of cell types. How can I find out if my cells will work on this platform?
Yeah, that's a great question. So we have, as I said in the webinar, we've tested over 80 cell lines. If there's a line that you're interested in, we are more than happy to engage with you to talk about your particular applications and your workflow. We've tested a really wide range of adherent, suspension, primary cells, and that information is freely available. But also, we're always looking to expand that list, and we really enjoy engaging with potential customers and current customers to help them develop their workflows on our system. We currently are running a clone challenge where users who are struggling to develop cell lines in their own labs can submit for entry, and then we will actually help them internally at our site to develop clones for their workflow so that we can show them how to leverage the AIR system for their own experiments. And you can enter to be selected for that on our website. So you can just go to cellmicrosystems.com and a banner will pop up showing you how to get involved in the clone and into the clone challenge.
Thank you. So another question here is, have you tried adherent HEC 293 cells in serum-free mediums such as Freestyle 293?
Yeah, so adherent HEC 293 T cells grow really beautifully on the system. The time from single cell seeding to clonal formation is very short for these cells. Typically from single seeding to colony isolation is around 3 days. We have actually done freestyle media successfully without the serum addition, but you can also add additional serum additives or serum replacements if you wish in order to increase isolation efficiency. We've also done this with the suspension HEC 293 freestyles and other suspension 293s like the XP cells. And we actually have a RAF note on our website that covers some of the data showing how the success rate of the 293 freestyles compares to other adherent cell types. So if you're interested in more information on that, you could certainly download the RAF note from our website.
Thank you. And how long does it take to scan and to isolate?
Great question. It's incredibly rapid. So for our typical bright field only scans on our quad array consumable, a typical scan takes about 6 minutes to complete. And that's really quite astonishing when you think about the fact that it's capturing every single field of view and every single cell raft every single time. So it's really very rapid. It allows you to do very quick scans of your consumable and then put the consumable right back into a standard tissue culture incubator without actually tying up the instrument for other users. So it's really good for large labs or core labs in that regard, because unlike a live cell microscope or a sorter where you're monopolizing it for a single experiment, you can just do that quick time-based scan, 6 to 10 minutes if you're adding on fluorescence, and then put your consumable right back into the incubator to keep your cells and in an incubated environment and not take up time on the instrumentation.
Okay, thank you. And another question I've got here is, what's the method to transfer the microwell from CellRaft to 96 wells for expanding each clone?
I guess this question is probably asking about once the clone has grown off the raft. That's actually very easy. It's just like any other normal passaging that you would do for tissue culture. So once that magnetic wand has deposited your cell raft in the microwell plate, the cells can grow off of the raft passively until it reaches a normal size for typical clonal passaging. So at that point, you would trypsinize and split your cells just like any other cell line that you're used to doing in regular, flask or plate-based culture. With the suspension cells, it's even easier. Once they have been transferred over, they will usually grow attached to the polystyrene cell raft for a few days. And then with a simple trituration, they'll go right back into suspension in the 96 well plate, and they could be passaged as normal. So it's really quite straightforward. And because your clone is already established, once you've isolated the raft, rather than isolating a single cell, your time to expansion is much shorter. So we typically find that the cells have grown off raft in about 3 days, usually up to about a week, and then they're ready for passaging and expansion into your working cell bank or for further characterization in whatever your downstream assay is.
Thank you. And another question I've got here is, have you used this technology for screening primary immune cells, for example, antigen-specific B cells?
Yes, actually, we have. So we have some literature published showing that we have done this with primary human T cells, and we've developed some assays for T cell killing, such as CD8 T cell killing and CD4 T cell proliferation. And we do have some work that we've been doing with primary B cells, although we didn't talk about it today, and we're more than happy to engage customers to talk about how their workflows could be used to screen antigen-specific B cells.
Thank you. And we've just got time for one last question, and that is there an on-stage incubation?
So the instrument in its most entry-level configuration does not include on-stage incubation because of the rapid scan times and rapid isolation times. We don't find that most customers need the incubation or CO2 or environmental control. If you are working with particularly long experiments or really sensitive cells and you're concerned about it, you can upfit the instrument with an on-stage incubation that will include temperature, humidity, and CO2.
Okay, thank you. And I think that's all we've got time for today. So I would like to thank our expert speakers for today's informative presentation and discussion. And a big thank you to everyone joining us online. We hope that you found this session worthwhile. So if you have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speakers. Remember, you can also download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.